Welcome back. Let us discuss the role of nucleus in cellular programming. Welcome to another episode where we discuss within the nucleus. We are a team of two people. I'm Sukant Khurana, and with me is Basudha Banerjee, a brilliant student of regenerative medicine. We are here to discuss how nucleus can shape our future. Here we dive into the fascinating world of regenerative medicine. Today we are exploring a fundamental topic, the role of nucleus in cellular programming. Joining me once again is Vasudha Banerjee. And welcome, Vasudha.
Thank you for having me, and I'm excited to discuss this topic with you today.
Yeah, let's start with the basics. Can you explain cellular reprogramming and why is it so important in regenerative medicine?
Absolutely. So, cellular reprogramming is the process of converting one type of cell into another by altering its gene expression pattern. And this is extremely crucial in regenerative medicine because it allows us to create induced pluripotent stem cells, which are iPSCs, from adult cells. And these iPSCs can then be used to generate various types of tissues, which offers immense potential for repairing damaged organs and treating diseases.
Well, that's fascinating. And how does the nucleus play a role in this process?
So the nucleus is the command center of the cell, housing all our genetic material. It controls the gene expression, which determines a cell's identity and function. And during reprogramming, the nucleus undergoes significant changes. Specific factors are introduced to revert the cell to a pluripotent state, essentially resetting its entire genetic program. This involves modifying the epigenetic landscape, the chemical tags on the DNA, and histones that regulate its entire gene activity.
So the nucleus isn't just passive storage unit for DNA. It actively manages how genes are expressed.
Exactly. The nucleus is dynamic and responsive. It integrates signals from the environment and other parts of the cell to fine tune its gene expression. This plasticity is what makes reprogramming possible. And by manipulating the nucleus, we can coax cells into adopting different and new identities, which is the basic foundation for many regenerative therapies.
Can you give me can you give us an example of how this reprogramming process works in practice?
Sure. So one of the most well-known examples is the creation of induced pluripotent stem cells using Yamanaka factors. So these Yamanaka factors are basically four specific transcription factors, OCT4, SOX2, KLF4, and c-MYC, that when introduced into adult cells can revert them to a pluripotent state. The nucleus responds to these factors by altering its chromatin structure and gene expression patterns and effectively turning back the clock on the cell's developmental stage.
And what are the implications of this for regenerative medicine?
See, the implications are quite vast. iPSCs can be differentiated into virtually any cell type in the body, making them a very powerful tool for tissue engineering and organ regeneration. For instance, they can be used to create new heart muscle cells for patients with cardiac disease or even neurons for those with neurodegenerative conditions. Most importantly, because iPSCs can be derived from a patient's own cells, they also reduce the risk of immune rejection, which is a very significant challenge in traditional organ transplants.
That's incredible. But I imagine there are also challenges associated with this technology.
Yes, there are several challenges. One major concern is ensuring the safety and stability of these reprogrammed cells. The process of reprogramming can sometimes lead to genetic abnormalities or even epigenetic memory that can affect the cell's behavior. Moreover, the efficiency of reprogramming can vary, like not all cells respond equally well to the process. So researchers are continuously working to optimize these techniques to make them more reliable and more safe for the clinical translation.
Yeah, it sounds like there is still a lot to learn and improve upon. What are some of the current research directions in the field?
The current research is focused on several key areas. So one of them is improving the efficiency and safety of reprogramming methods. Scientists are exploring new combinations of transcription factors and even small molecules that can enhance the process. Another area is understanding the epigenetic mechanism that governs the cell identity and thus how they can be manipulated more precisely. Additionally, there is a lot of interest in developing better culture conditions and biomaterial scaffolds that support the growth and differentiation of iPSCs into functional tissues.
It's clear that the nucleus holds the key to many advancements in regenerative medicine. Thank you, Basudha, for sharing your expertise with us today.
My pleasure. It is an exciting time in the field and I'm glad we could discuss it.
That's all for today's episode of Within the Nucleus. Stay tuned for more insights into the world of regenerative medicine. Until the next time, stay curious and keep exploring the wonders of cellular science.